I'm Noelle.
I'm Derek Yu.
And we are testing the regenerative effects of protein FSTL1 and coenzyme Q10 on spheroid and 3D bioprinted myocardial tissue patch models post-injury. So for a little bit of background before we begin, cardiovascular disease is and has been for the past century the #1 killer globally. And the reason as to why cardiovascular disease is such a global killer is because of the fact that heart muscle cells, also known as cardiomyocytes, cannot proliferate post-injury. So this means that any sort of muscle tissue damage that happens within the heart is replaced by scar tissue. So every time the heart muscle deteriorates, there's just a replacement of this muscle tissue with scar tissue, which causes the heart to grow weaker and weaker after every single injury. So essentially, heart muscle damage is irreversible. So we are focusing on these specific issues of the non-proliferative characteristics of cardiomyocytes in mammals. So in all mammals, cardiomyocytes cannot proliferate post-injury. And another specific issue we're focusing on is model efficacy. So in vivo models are very costly, and this is essentially when we use animal testing like mice or vertebrate models to test different treatments. And we wanted to find a middle ground between the 2D cell culture model and in vivo models, as 2D cell cultures are often oversimplified. So 3D printing and 3D cell cultures allowed us to do this, and Derek will talk a little bit more about this.
So as Noah mentioned, our project has two main goals. The first goal is to evaluate the effects of protein FSTL1, or follistatin-like 1, and coenzyme Q10 on stimulating cardiomyocyte proliferation. And protein FSTL1 is a protein that's been shown to have stimulative effects on cardiomyocyte proliferation, and coenzyme Q10 as an antioxidant is known for having a cardioprotective role. The second part of our project is to develop a 3D bioprinted patch to provide a compromise between the affordable cost of 2D cultures as well as the complexity of in vivo models, as Noelle mentioned before. So we're going to briefly go over our methodology and some challenges we faced. So our protocol was overall pretty simple. For spheroids, we essentially cultured cardiomyocyte cells, then turned them into spheroids, which are basically balls of cells. Then we inflicted an injury type called ischemia reperfusion injury. Then we imposed our treatments, FSTL1 and CoQ10, and then evaluated our cell viability of each group with a trypan blue stain. And for the patch, we did a similar process where we cultured HNC2s. We loaded them into a gelatin methacrylate-based bioink, and we then printed with a bioprinter. And the next steps we would have taken, which we wanted to do if we had more time, we would have also afflicted ischemia reperfusion injury and post-treatments and evaluated cell viability.
In terms of our spheroids, which is the hanging drop technique that Derek briefly mentioned, we did culture over 200 spheroids throughout our experimentation, and we were able to collect some pre-hypoxia control data. So as Derek mentioned, the injury that we are focusing on is ischemia reperfusion injury. So this is essentially when hypoxic injury is induced, so oxygen is deprived from our cells, and then oxygen is reperfused back to the cells. So mimicking what happens within the heart when the heart tissue is deteriorating because of lack of oxygen, and then blood flow brings that oxygen back in. And we found that there was a 62.45% reduction in cell viability, which showed that the ischemia reperfusion injury that we had imposed was working. And we also focused on a few other groups. So there was our no treatment control, which was the viability testing that we did to determine if the ischemia reperfusion injury was working, and our FSTL1 individual treatment group and our coenzyme Q10 individual treatment group. So as for the 3D models aspect of our project, as you can see, there are 3 diagrams that can be seen on the slide. And so first, in figure 2.8, you can see a 2D H9C2 cardiomyocyte cell culture. So we saw that our cells were very healthy and happy and growing inside of our cell flask. So you can see that the cells are adhered, and this is a standard 2D cell culture. In comparison, you can see in figure 2.B, our H9C2 cardiomyocytes are in a spheroid formation. And so this is when all the cells have kind of clumped together due to gravity pulling it down, and it's in its droplet form. So that spheroid can be seen to also be viable. And last but not least is our H9C2 cardiomyocytes inside of the 3D bioprinted gelatin methacrylate patch. And so as you can see, the cells are a bit more rounded because post-trypsinization, we allowed the cells to be diluted with media, and the cells were kind of free floating inside of the patch. But we were really excited to see this because it was indicating that we were able to successfully bioprint a viable myocardial tissue patch. As for our test prints, as we were working towards our goal of printing the gelatin methacrylate patch, we went through a series of steps of different types of prints using different materials to test our structure that we were aiming for, which is this pi lattice structure that you can see over here. So first we tried a PCL patch test print, and PCL is just a type of plastic, just using the 3D bioprinter to see what kind of structure we were aiming for and if it would work, and as you can see, it did work. Then we use Pluronic, which has a viscosity and build that's much more similar to the gelatin methacrylate. So we use Pluronic F127 for our monolayer patch test print. And finally, we use the gelatin methacrylate to create our cell-laden gelatin methacrylate patch.
So we faced a lot of challenges while trying to get our gelatin methacrylate to print. And to address these, we varied a lot of different print settings. We adjusted pressure, which is the PSI with which the bioprinter pushes out bioink. We adjusted our needle gauges, which is the size of the needle of the syringe. We adjusted our temperature settings, which is just print temperature. And there's a lot of different reasons for why a lot of the times we face difficulties trying to get it to print. Sometimes it would cross-link prematurely if we didn't properly protect it from ambient light. Other times there's different frictional differences between the bioink and the sides of the syringe. And also the syringe was sometimes just straight up clogged or bent. And so here you can see some of the pictures of our gelatin methacrylate after a non-ideal print attempt. And over here, you can see even more of those unsuccessful attempts. Sometimes the gelatin methacrylate would barely come out at all. Other times it would just kind of splutter out all at once. And both of those scenarios are not ideal.
So some solutions that we used to combat these challenges Derek mentioned was first that we prepared the gelatin methacrylate in a low light environment. So we did this through wrapping this container that the actual gelatin methacrylate was prepared in foil so that the gelatin methacrylate would not cross-link prematurely. We also switched to a plastic syringe and placed a stopper in it during our print, which allowed for even application of pressure despite uneven friction. So this would account for this kind of spluttering or clogged aspect of our print. And lastly, we put the gelma in an ice bath for 10 minutes before printing so that the viscosity would change and the gelma would become a little bit more solidified so that it wouldn't drip out of the syringe. So as for our results, quantitative analysis has shown us, as mentioned before, our pre-hypoxia, post-hypoxia data shows that the ischemia reperfusion injury that we were imposing on our cells was successful. And we were also able to get some cell viability percentages for each of our treatment groups within our spheroid data. So as seen in this table, you can see that our FSTL1 treatment group and coenzyme Q10 have much higher average cell viability percentages as compared to our no treatment group. So with these additional treatment factors, there seems to be some sort of change. And based on this just graphical analysis, this difference can clearly be depicted, but Derek can talk a little bit more about our statistical testing.
So we used a Mann-Whitney U-statistics test to compare the cell viabilities of our FSTOL treatment groups with our no treatment groups, as well as the coenzyme Q10 treatment group with our no treatment group. And so after performing this test, we found that our FSTO1 treatment group actually did show a statistically significant difference between the cell viability when compared to the no treatment group, whereas coenzyme Q10 did not. However, as you can see based on this bar graph, the average cell viability between the protein FSTO1 group and the coenzyme Q10 group was very similar. So we believe that the fact that one of them is statistically significant while the other isn't is more likely due to a small sample size than an actual issue with the coenzyme Q10. So continuation of this research would involve simply just collecting more data to try to reduce the impact of outliers. So we did find that FSTO1 treatment did show statistically significant improvement in cardiomyocyte proliferation, while our coenzyme Q10 data was inconclusive. So as I just mentioned, the immediate future continuation of this research would be to, we would want to test our combinatorial treatments, which we did not actually get to. Basically, we would want to test protein FSTO1 combined with coenzyme Q10. We also want to perfect our printing protocol because, as you see, we had many different failed attempts, and that reduced our ability to actually collect cell viability data on our patch, and we want to be able to do that in the future. Some potential therapeutic applications of this research is particularly with regard to custom printing myocardial patches to help treat ischemic heart disease on a patient-by-patient basis. Because the thing with these patches is that you can customize the structure, and by doing so, we can dramatically improve the effectiveness of cardiovascular treatment. Initially, we have, of course, protein FSTO1 as a potential regenerative treatment to heart disease. Long-term goals and possibilities for bioprinting involve increasing the organ bioavailability for use in organ transplants by actually circumventing our current reliance on organ donors by potentially bioprinting entirely new vital organs in a laboratory.
Thank you.
Thank you.
All right, we have about 4 minutes if there are any questions for our young researchers.